Okay, welcome back. I don't know what you guys got. I got some water. I was parched. Anyways, let me see. We're going to talk about the cytoskeleton. We're going to talk about the plant cell and consequently photosynthesis again. And we're going to talk a little bit about cell communication. Okay, so that's what we're going to converse about this time. Excuse me as I try to move this out of the way. I can't. Okay, fine. So, the cytoskeleton, as the name implies, is internal scaffolding. It provides the cell with structure, facilitates the movement of materials inside the cell, and facilitates cell movement. Okay. Um, let's see, there are three principal types of cytoskeletons. You should know this. Uh, Going from smallest to largest, microfilaments, intermediate filaments, and microtubules. Okay. That's kind of what they look like. You should appreciate these photographs. These are very cool photographs. In case you don't know what's going on here, these are fluorescently fluorescently labeled uh, cells. What they do is they take antibodies that are specific to different proteins. And this one, of course, it would be actin. This one would be lamins. This one would be tubulin. And then they put a label on those antibodies, and it lets you see different aspects of the in this case the cytoskeleton. Very, very cool. So here's the microfilaments, as this thing just prevented you from seeing. I put it on the bottom. Microfilaments in red. See that? These are the tiniest one. You should know that they're made out of actin. Okay. These are intermediate filaments. These are right in the middle, obviously in green. And we're not going to ask you to remember what these are made out of because it's kind of a long laundry list. Uh, some of them are called lamins. Uh, some are called valins. You don't need to remember these, but you do need to remember that this is actin. Okay, and you do need to remember that for microtubules is made out of alpha and beta tubulin, or just tubulin. Okay, you can see that it's alpha and beta because it's got two different colors. Do you see that? So, let's see. I'm going to go with the author. That way I don't forget to say something. Okay. Microfilaments are made of the protein actin. I think we already covered that. Now, this is interesting because in you and me, as we move, our muscles have two main proteins. One is actin and the other one is myosin. And cells use microfilaments, one of the components that we use in muscle, to move themselves. Okay, so that's actin. They help the cell move and capture prey by forming rapidly in the direction of movement and decomposing rapidly on the other end. So here they show a human monocyte going after this poor defenseless E. coli over here. And you can see that that monocyte is making this huge actin pseudopod, is what it's called. It's called a false foot, a pseudopod. And then that pseudopod will move around this bacterium and it will engulf it. That's called phagocytosis. Okay, and is going to join it to, not surprisingly, a lysosome, and that lysosome is going to turn into a phagolysosome, and then that bacteria is going to be turned into a whole bunch of nothing. It's going to turn into amino acids. It's going to turn into fatty acids. It's going to turn into uh, recycled materials that the cell can use. Is that incredible? Now, this is a white blood cell, so probably what it's going to do is some of it is going to store and do what's called antigen presentation, but that's another story. So now, before I move on to intermediate filaments, this gives me an opportunity to talk about something very, very cool. Let's see if I can. Hello. Oh, there we go. So I brought up an amoeba, okay? Because that leukocyte, that white blood cell that we just looked at, it moves through your tissues using what's called amoeboid motion. And effectively, what it does is it pushes its microfilament made of actin to make a pseudopod. So this amoeba has 1, 2, 3, 4, 5, 6, 7, 8 pseudopods, right? And as it's pushing out, because there's no real shape to this cell, the rest of its cytoplasm, not just the cytosol, the cytoplasm, that means everything inside of it just gets pushed in that direction. And we call that cytoplasmic streaming, which is so cool. This is lovely to see under a microscope. Let's see if I can get this guy. Look at that. See the actin forming and see the cytoplasmic streaming taking place there? Is that awesome? Look at that. This is real time. This is how fast these things move. See? The cytoplasm is moving from where it's coming from as this guy comes in and is moving towards where it's going to as it looks for food. Is that awesome? Oh, that's unfortunate. That was fun. I need to cancel that before something weird comes up. Okay. So cytoplasmic streaming, there's something else I want to say about that. Oh, amoebas are fun to play with. You can have your own pet amoeba. You should be careful because they're not all nice, you know. It's like dogs, some of them bite. But you know, if you wear gloves, you can play with them. And what you can do is you can feed them. So you can grab cornstarch, add food coloring to it, red or yellow or purple or whatever, and then you put a little bit of cornstarch with food coloring into the plate with the amoeba, and the amoeba will eat the food starch, and then it'll have vacuoles that have the same color that you fed it. So it'll have purple vacuoles or red vacuoles. Um, Typically, if you find an amoeba with colored vacuoles, you run into somebody who doesn't have a life. I shouldn't say it that way. That's terrible, isn't it? I just suggested that you do it. Well, this is what you would do under COVID-19. I mean, you're at home, you can't go out. Feed your amoeba, right? It'll be okay. All right, let's go to the next one. Intermediate filaments. So intermediate filaments provide support and structure to the cell. It is the least dynamic. In other words, they don't move at all. So if you look at a cell and you're like, you know, is this strange, or does every organelle in the cell look like it's always in the same spot? Well, it's not strange. Every organelle in a cell is in the same spot. At least not the amoeba. And the amoeba is a bad example, but let's say you have a muscle cell or a nerve cell. Okay, you look at the organelles and they do not move. They are very stationary. And the reason that they're stationary is because you have intermediate filaments holding them in place. This is where the mitochondria are going to be. This is where the rough endoplasmic reticulum is going to be. This is where the Golgi complex is going to be. This is where, I mean, it is right there. So we say that it is the intermediate filaments is the one that is most similar to our own skeleton because it doesn't move. Least dynamic. Very, very cool. Microtubules play a structural role in cells and facilitate the movement of materials inside the cell by serving as transport rails. Microtubules are formed out of tubulin. So this is inside the cell. Outside the cell, microtubules form cilia and flagella. Okay, and both cilia and flagella can be used for moving things into the cell, or moving the cell in general. So cilia stem from cells in great numbers, serving to move the cell or to move materials around the cell. Oh, this is making me sad because I didn't bring up a rotifer. But you know what? I've got the technology. I bet I can do it. Let's get out of here. Let's see. Let's look up rotifer. Let's look at a cute little rotifer here. Hurry, 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 hurry. We're going to run out of time. What do you think? No, that guy's moving too fast. We need a slower rotifer. Let's try this one. Let's see what it does. I should have queued it ahead of time, but now that's not showing what I wanted to. So I don't know if you can tell, but you can see a stream of fluid moving through here. You see the cilia moving right there? That's incredible. So it's got cilia, and that cilia is going. And this guy he's at a buffet, right? He's just eating. Look at that cilia move right in his little mouth. Look at that. So it's like, this could be breakfast, right? So you can see how cilia can function in these guys to eat. Very, very cool. Okay, but I digress. So cilia stand from cells in great numbers and serve to move the cell or to move material around the cell, or as you saw, to feed a cell, right? When cilia cease to function or damage due to tobacco smoke, the ciliated pseudostratified layer of our bronchioles is unable to clear debris from our lungs, and it yields the so-called smoker's cough, okay? That's my PSA for the day. If you want nice, healthy cilia and your pseudostratified epithelium, stop smoking so that they're healthy and they can clear the garbage. Your lungs is an organ that's azenic. Azenic, that's a word. Azenic means that it is free of foreign contaminants. It is free of debris. It is free of microorganisms. And the reason it's free of external debris is because you have this pseudostratified epithelium with cilia, with mucus on top of it. And as you take every breath in, the garbage that you breathe in sticks to the mucus, and then the cilia pull it back out, and it goes into your throat, and you swallow it. Yep, exactly what I said. We all swallow about a pint of mucus a day. Keep on watching this, and you're never going to want to kiss anybody. Trust me. But it's healthy, okay? So that mucus really is just glycoproteins that are protecting your very healthy lungs from this garbage, right? And then as you swallow it, it goes into your stomach, where the hydrochloric acid in your stomach takes this bacteria and destroys it and protects your beautiful lungs from it, okay? But all this can happen only if you take care of your beautiful pseudostratified epithelium in your lungs, okay? So all this about microtubules. Let's see, flagella, in contrast to cilia, is not many. Flagella is only one, okay? And its main function is cell movement. We're doing okay with that? So flagella and cilia, the main common function is cell movement. And then this is tubulin. So this is very cool. Notice that tubulin, here's cilia, here's a flagellum, and here's A and B tubulin, making transport monorails inside your cells. Okay, so imagine that you have the rough endoplasmic reticulum and the Golgi complex up here. Okay, what's causing these vacuoles to move from one point to the other is the fact that we have this monorail coming out here made out of tubulin, and we have these motor proteins moving the vacuoles in one direction. Okay, there's your transport vacuole. So it goes from the rough endoplasmic reticulum to the Golgi complex, and then to the plasma membrane. Okay. Hey, what happens if we have to go in the opposite direction? You need a different monorail to go in the opposite direction. So we have quality control here, and we decided that this vacuole is going the wrong direction. This needs to be transferred to a monorail that goes in the opposite direction, where it'll meet a lysosome and be destroyed. Okay. So the function of tubulin is much. Okay. Starts right here. Microtubules made from tubulin. Cilia and flagella is made from microtubulin. Okay, we learned the purpose of cilia in the lungs and the fact that both cilia and flagella are used for the movement of cells. Okay, so microtubule, very, very important. Okay, the plant cell. One thing that's interesting about plant cells do not have lysosomes. And the reason they don't have lysosomes is because they have a central vacuole that takes over the role of the lysosome. Okay. Otherwise, they have ribosomes, they have a cell nucleus, they have a rough endoplasmic reticulum, everything. Okay. Three structures not found in animal cells or in plant cells. That's the cell wall. Okay. This is very very thick and very sturdy and not pliant at all, not like a plasma membrane. Okay. It has a large central vacuole, and it has an additional organelle called a chloroplast for photosynthesis. So here's a cell wall. Okay. Here's a central vacuole. Remember that central vacuole degrades material. It's a place for the storage of nutrients. And another thing that I haven't seen on this slide is it provides plant turgor. So you know this, when you have your little tomato plant at home and you forget to water it and it wilts, okay, your plant no longer has turgor. So you add water, and then after a while that poor thing comes back to life and your little leaves get all nice and tight. Okay, that central vacuole has now pushed the cell wall out and has increased plant turgor, so that your plant is nice and stiff the way that it's supposed to be. Okay, so that's another thing that it does. Here's her chloroplasts, right? The photosynthetic organelle. Do you remember why this is cool? It has its own DNA. It's part of the endosymbiotic theory. So both mitochondria and chloroplasts are known to have been engulfed by eukaryotic cells. Very, very cool. Thank God not us, right? Thank God not us. Can you picture yourself walking down the street photosynthesizing? Or you're inside and you're like, hey, Sally, do you want to have breakfast? Nah, that's okay. I'm going to go outside and photosynthesize. And then we'd have all like a green sheen to our skin because we all had chloroplasts. Wonder what made evolution decide that mammals would not have chloroplasts. Whew, that's probably a close one, huh? Okay. So cell wall gives the plant structural strength and helps regulate the intake and retention of water, right? We go back to turgor, which is the pressure provided by the water as it comes up. Chloroplast and energy. So just like mitochondria are involved with providing energy, chloroplasts are involved with the harvesting of energy. Okay. Chloroplasts are the sites of photosynthesis. In case you forgot, I think last chapter we said that photosynthesis and respiration were exact opposites of each other. So here's photosynthesis. You take 6 molecules of carbon dioxide, you take 6 molecules of water, you take some sunlight, and then this beautiful chloroplast gives us a delicious molecule of sugar and 6 wonderful molecules of oxygen. You and I are alive because of chloroplast. If it wasn't for chloroplast producing delicious sugar and delicious oxygen all day long, you and I could not eat plants which make sugar, or eat delicious little cows that eat sugar, right? Or little goats, or whichever little four-legged animal you like to eat, okay? Because they all depend on grasses or hay, something that produces sugar. And these chloroplasts produce oxygen for you and I to breathe. And then, as if that wasn't enough, they take away carbon dioxide from our atmosphere. Remember, that's bad because we have too much carbon dioxide. So they take carbon dioxide, water, and the sun's energy. Okay, what is that? This is physics. This is a photon of light, and it's h nu. You don't need to know that. And then they give off yummy sugar and yummy oxygen. Don't forget this. This is important. There's nothing more important. This is why you and I are alive, because chloroplasts produce sugar and oxygen. If we didn't have chloroplasts, you and I would not be anything but a dream. Do you remember that this is the opposite of respiration? In respiration, you and I consume sugar, we breathe in oxygen, and then we exhale water, we exhale carbon dioxide, and we produce adenosine triphosphate, about 36 of them, okay? Cell-to-cell communication, this is the last topic. Okay, plant cells have channels called plasmodesmata that are always open and hence have the effect of making the cytoplasm of one plant cell continuous with that of another. Why is this important? Because really we need tissues and plants to communicate with each other. And this is what, they don't have a nervous system. So this is another way for them to communicate, is through these channels called plasmodesmata. You and I have the same thing, except that with us, they're called gap junctions. Why are they different? Because plant cells have a cell wall, you and I don't. So plasmodesmata are a little bit more complicated than gap junctions, okay? But same concept, protein assemblages that open only as necessary, okay? They allow cells to communicate with each other. Look at the width here. Look at the width of the plasmodesmata, because this is a cell wall and this is a cell wall. See the width there? That's quite a little job there. Also, that these cells can communicate with each other. By contrast, look at the width of our gap junction. Here's one plasma membrane. Here's another plasma membrane, okay? And if you can't think of a good example, I'll give you one. Think of your heart, okay? Your heart has a pacemaker site, and then it has an SA site, sinoatrial site, and then it has your Purkinje fibers, and then it has its, it has this entire electrical complex, okay? And if it wasn't for the gap junctions in the cells of the heart, it would not be able to beat rhythmically. Does that make sense? So it's important for many cells in much of our body to be able to communicate with each other through ions electrically, okay? And again, in our body, we use gap junctions. Okay, that is it. I hope you survived this chapter. I hope I didn't bore you too much. And have yourselves a wonderful rest of the day if I don't see you again, okay?